let's dive into that presentation. This is Special Senses Part 2. Okay, hold on. All right, let me go back up to this part, to the beginning. Again, ladies and gentlemen, one of the questions in your exam is, it's approximately 70%. 70% of the special senses is the eye. Remember, we talked about that. How much data is given to the eye, processing to the eye. So that's why we are covering a large content of the eye, just to get you back on track. I will ask you some questions in terms of light, refraction. There's some clinicals. And I promise you, I went through the entire test bank. I didn't just like click stuff, like a madman. I went very carefully looking in there, making sure to do my best. Again, there may be some that I have made some that may not fully reflect exactly things that I have said and covered, but for the most part, I've done my very best to make darn sure that that is the case. The last part that I want to talk about is mainly focusing on light and its optics. Let's talk a little bit about wavelengths and colors, and we're going to get into what they call color blindness, right? Or what's the other word for color blindness? Like, I don't know, color deficient or something like that. So electromagnetic radiation, that is what in terms of that we have in wavelengths and light. So it's a form of energy. Remember, energy has different forms. This is not the end all be all of energy. Okay, so these electromagnetic radiation, they have energy waves. Okay, so from a long radio waves to short X-rays. Some of these wavelengths, they're measured in nanometers. And light has wavelengths between 400 and 700 nanometers. However, the eye can only respond to visible light, meaning we can only see what we're supposed to see, right? Like that's all we can. It doesn't mean it's not there, like infrared, ultraviolet. We can't see those things. They're there, they're present. They do have its signature, but we can't see that. I'm not going to ask you to know this. I'm just showing you that there are different ranges in terms of wavelengths, right? Please just keep that in mind. Now, I'm just gonna jump here real quick since we were talking about wavelengths. So we go here, wavelengths, we go here. This is the visible spectrum. You have 400 nanometers to 700, as was indicated here, light, okay? And you have visible light, okay? So as a result, you have, say, these are known as at 420, it says blue cones, rods, green cones, red cones, and these are the associated wavelengths to that. Let's go back this way. Light, it's packages of energy, photons that travel in a wave-like fashion in high speeds, okay? In high speeds, so they move in this direction. Visible light passes through a spectrum and it's broken up into bands of color like a rainbow, right? So they're moving, they have a different pitch, a different width. So red wavelengths are the longest, have the lowest energy. The color that eye perceives in refraction of a wavelength. So grass is green, because it absorbs all colors except green. And as a result, it is reflecting off the non-absorbed wavelength, is reflecting back, and that wavelength has a signature on it, which then we perceive as green. White. White reflects all colors and black absorbs all colors. Very interesting stuff in terms of what we actually see. Please know this term. I will ask this term of you in terms of bending of light rays, refraction. It is in your exam. Refraction. So due to change in speed of light when it passes from one transparent medium to another, and path of light is like an oblique angle. So it has these, it refracts. So for example, convex, concave, well, that means lenses of eye can also reflect light because they are curved on both sides. That is actually what is supposed to happen. So convex, thicker in center than at edges, concave, thicker at edges than in center. This is an example of a refraction. I think all of us have had some experience with this. You can see the refraction of light. The image that you see under the water path from this level to this level is inaccurate. It is not true. That is not what is reality. The path should continue for this way over here, but it does not, because water has resulted in the refraction of light. This is why it is difficult and it takes training to spear a fish. What you see, and you have a spear and you see a fish and you throw the spear to obtain the fish and harvest it, it is usually not where your eye sees the fish because of refraction, okay? Light and optics, refraction and lenses, convex lenses bend light passing through. Make sure we know that. So light can converge at a focal point. So we want to funnel this light in. We want to have a focal point. Image formed at focal point is upside down and reversed from left to right. So the concave lenses disperse light, preventing light from being focused. Opposites, right? You see this? Convex, rays converge at a focal point. Concave disperse light, preventing light from being focused. So please know the difference between convex, concave. Also, know this path. I ask of you in the exam, the pathway of light entering your eye, okay? So make sure we understand where we're going from: the cornea, aqueous humor, lens, we have vitreous humor, intraneural level retina, and finally in the back, those photoreceptors. Remember those photoreceptors we talked about? So know your path, because I have a question in there and I move these around for you. So make sure you know your path. Light is refracted 3 times along this path, all right? It is refracted entering the cornea, entering lenses and leaving lenses. 3 times it is refracted. Now, majority of refractory power is in the cornea. However, it is consistent and cannot change focus. So with this in mind, lenses is able to adjust its curvature to allow for fine focusing. This is what we were talking about earlier. I was talking to you about that the eye is highly responsible for focusing. And I was trying to explain to you that depending on what your life is and how your lifestyle is, meaning the things in your life are close, if you have a job that requires further distancing, it can impact the state of your eye. So as a result, it says focusing light on retina. It can focus for distant vision and for close vision. It is capable of doing that. Sometimes we get into some problems, right? So focusing for distant and close vision. Look at this. So we have ciliary muscle, lens, and we have this suspensory ligament about here. For focusing for distant and close vision, ciliary muscle, the ciliary muscle and ciliary zonal focus an image by changing the shape. It has to change the shape because it's just like you moving binoculars. You're using your microscope in lab. And don't forget about that, the arrangement of that sphincter-like around the lenses. So ciliary muscle contraction loosens the ciliary zonal fibers and relaxation tightens them. So that makes sense. So a relaxation, it's going to loosen, contraction tightens. So focusing for a distant vision, eyes are best adapted for a distant vision, not for close up vision. Again, a topic that we discussed the last time. But the world that most people of today, at least in our country, we're not using a lot of distant vision. But this makes perfect sense going back again to Mark's input that he did the first time. The input was survival. That is the word he used. So it makes total sense that the eye is best adapted for distant vision than close vision. What in the world would you want to see really super up close when you're out in the brush, right? And there are animals faster, stronger, and bigger than you. It makes no sense. None whatsoever. So this makes sense to have distance vision. Far point of vision, distance beyond which no change in lens's shape is needed for focusing, right? Now look, 20 feet, normal eye. Then we get into the whole 20/20, right? Corneal lenses focus light precisely on retina at a distance. So as a result, we look back at this one here, and here we have it. Ciliary muscles are completely relaxed in the distance vision. Of course they're relaxed in the distance vision. Why the heck would you want the ciliary muscles to be all tight the whole entire time of a person's life who's out in the brush? Why? Why would you want that? To have that type of contraction. Not only that, you're gonna have that type of contraction, you're gonna have to be deploying ATP to do the contraction, adenosine triphosphate, the energy required to make that type of action. No, it doesn't make any sense. So ciliary action is completely relaxed in the distance vision. This is some of what we're, last time I talked to you about sympathetic involvement. So here we have sympathetic input relaxes ciliary muscle, right? So we go back and we realize that the other chapters that we have covered are coming back now and having a little role here. Here we just have this here. You can see the direction moving, and in this right here was relaxed ciliary, tightened ciliary, flattened ciliary. And this all has to do with in terms of looking back and going and say, okay, this makes sense for the human body to see at a distance. Now, focusing for close vision. Well, we're going to have to do some adjustments, because if the eye is naturally adapted for a distant vision, then we must change something for the close vision. So eye requires active adjustments using simultaneous processes. And these are the processes. So accommodation of the lenses, you have to change the shape to increase the refraction. Near point of vision, closest point in which the eye can focus. And then the presbyopia, loss of accommodation over age of 50. Contraction of pupils, conversion of eyeballs. So all these are very, very, very important in terms of, we're talking about light, the divergent pathway of light entering into the eye, right? So most of these questions I ask you. I also ask you clinicals on these, all right? Like myopia, all these, I ask you these clinicals. So again, I tried to do the best job I can in terms of covering content. So make sure you go over this point. This one's a very self-explanatory one. I won't get into this clinical because there's nothing I need to really truly explain to you guys. You guys have covered some very complex content in terms of nervous tissue, CNS, PNS, ANS, and now you've gone into the special senses. So these little words are no match for you guys. We will get into some other things once we get into the photoreceptors that I'll have to take a little bit more time to explain. Phototransduction, right? So when you think photoreceptors, I need you to think about that back of that eye, right? Think about that pathway of light, and I need you to think of rods and cones. Rods and cones, right? We've talked about rods and cones. We've talked about some of the differences between rods and cones. So in photoreceptors are modified neurons that resemble upside down epithelial cells, right? So of course, if they're going to be modified neurons, hey, come on, they're going to have a cell body, hello, a synaptic terminal and some two segments. So they are neurons. They're modified neurons, very unique, very special. We briefly went into this part, but and we kind of just a little bit like skimmed over it, or I dropped a few hints of it, in other words, in the previous presentation. But photoreceptors, very vulnerable to damage. We talked about detached retina. So they degenerate if there is a detached retina. It is important to try to protect your eyes from intensity, high light intensity, specifically like ultraviolet light and just different higher wavelengths, the wavelength signatures. Glasses are important. You can wear sunglasses that protect from UV light. All that is important because over time, it can damage the rods and cones. Vision is maintained because the outer segment is renewed every 24 hours. The tips of these photoreceptors fragment off and are phagocytized. A lot is going on there. And this is where it gets it gets complicated. Remember the direction, ladies and gentlemen, right? The eyes, the lenses are up here. So let's say up here and light is entering, and then we do have refraction, right? So we have to have refraction. We can't just go straight back, right? We have to get light, focus, 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 and spread it around so we can get the data. This is what we're looking at, right? It looks like an upside down epithelial, right? And we have our light. Our light is coming this way. So when we talked about that process of bipolar cells, right? So this one here, the blues are your rods. These are your cones. And well, if you have some sort of connection here, you have to have a synaptic terminals, right? You have to have a synaptic terminals. The body, this is the body, this is the body. The internal segment and the outer segment. Here are some disc. We won't get too much into these discs. We're not going to get too much in there. But these are the discs that do begin to have the process of being phagocytized. Again, these things take energy to do all this and do the pathway back, the information coming in, the information back. This is not cheap. This is not free. It comes at a cost. And the major cost and the currency of the human body is ATP, adenosine triphosphate. Therefore, there has to be some mitochondria somewhere. There just has to be. Here you can kind of see they try to help you out here by saying, okay, hey, look, there's some mitochondria in there because we're going to need some ATP to get this thing moving along. So rods are very sensitive to light, very sensitive. As a result, they are the ones best suited for night vision and your peripheral visions, right? Because if you're sensitive to light, then you can get information from sides. You don't need a lot of it. You don't need a lot of light intensity. You don't need a lot of data. So then you can do peripheral vision and you can do night vision. So those are one of the things that I wanted to mention to you. It does contain the single pigment, and as a result, vision is perceived in gray tones. Gray tones. And I mentioned something like that, I believe, in the exam. Now here, let's talk about cones. Rods, we said they're very sensitive to light. And here, cones have low sensitivity. Well, if they're going to have low sensitivity, they're going to require brighter light for its activation. Opposites, rods and cones, opposites. They do react quicker than the rods. And they have their 3 pigments. And they are responsible for high resolution vision. So these are some of the differences that we have. Now, some cones have their own ganglion cell. So the brain can put together accurate and high acuity resolution images. This is a good one. This one will help you in your exam. Feel free to use it. Table 15.1, the comparison of rods and cones, right? So just make it's nice and in order. Tables, that's the whole point of a table. It just put things organized, and you have this you have this resource right there for you. Well, we just talked about, hey, look at that. Rods, no color vision. Cones, color vision. Rods, highly sensitive. Cones, low light sensitivity. Rods, okay, low acuity. Cones, high acuity, right? Well, then we might have a place to say, where does color blindness occur, right? Where is this happening? Well, look, lack of one or more cone pigments, because it was the cone. It is the cone that is responsible for color vision. Just simple stuff, guys. Just, so keep that clinical in mind. I throw it in there for you. I want to talk about this one here. Please do not get this confused with the retina. This is retinal. It could be confused. Like in the quick glance of the eye, you may think it says retina. This is a very important light absorbing molecule that combines with one of 4 proteins to form visual pigments. This is synthesized from vitamin A. Okay, and I need to write up your lab case study. I'll get that done today. And I kind of held off because I'm like, wait a minute, I need to get into this last topic so I can give them as much tools to get this job done. Synthesized from vitamin A, retinol, synthesized from vitamin A. That means that you need this to make that. That's what that's saying. Okay. So just make sure that we get that, okay? That these 2 points are super, 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 super important. They're very, very, very important. Now, I just want to just briefly tell you, I will not get into the organic chem, specifically the subsection of organic chem known as stereochemistry. I'm not going to do that to you guys. This is just not the place for it. What I do want to say, and this is not in your exam, I do just want to briefly tell you what an isomer is, because you may encounter it in your medical field at some point. Now, it doesn't really do a very good job. Of course, you have to understand the background of the individuals who create the presentation. They bring up isomer, and then it says retinol is in a bent form in dark, but when pigment absorbs lights, it straightens out. Okay. So bent form is called 11-cis retina. Straight form is called all-trans retinal. A student may read that and be like, okay, what is an isomer? Well, what it means is that an isomer, they should put the definition there, it is a molecule that contains the same molecular formula, meaning it has all the same amount of atoms in there. However, its structure is different. At different times, it can change its structure. That is known as an isomer. So what's happening here is retinol, it says is in a bent form, meaning the structure retinol. They should say the structure retinol is in a bent form in dark, and they should show, I'm not going to get into the images. Actually, I've cut that out because I didn't want to get too in depth of that. They do show different images, but I just want to let you know what an isomer is. And then when the pigment absorbs, it straightens out, you see? So it's transforming. This never changed in terms of its atoms, it changed in its structure. Keep that in mind. And the reason why I'm telling you this is because if any of you are interested in nutrition, if any of you go down that path for yourself or anybody else, understanding of isomer is very important. Why? I just told you that the structures changed. I didn't say that the atoms were lost or gained or anything like that. I didn't say that. I said it stays the same, but it's the structure that changed under different environments or conditions. And I have trained you since AMP1 that structure leads to a function. I have constantly preached that to you, that structure leads to function. Well, the reason I'm taking a little time out of this, and this is not in your exam, but this is just a life lesson for those of you, most of you are pursuing that type of career in human health. I have told you before that vitamins that you buy from stores, right? First, they're not all FDA approved. I think most of us know that. Doesn't mean they're bad. It just means that some may be better than others. But what it really means is that you may not be getting the true values of the vitamins that are indicated in the bottle. One reason is because you will be excreting and urinating some of that out. Why? Because it is not bioabsorbed. Why? Because of the structure of the vitamin. So there are certain vitamins that they specifically design to have a specific type of structure, like vitamin E. I'm not going to mention the company's name, but there's a company that has done a lot of work on vitamin E, and I believe they market it. I take it, it's marketed as isomer E. So the average person gets it, looks at it like, oh, isomer E, junk. I don't, but they don't understand that. Whoa. What is that? Isomer. Now you know. And that vitamin E that I take has so much more bioavailability than the average vitamin E that you're going to find in a store. So you take those vitamin E's and you're not really getting the desired effect because it's not bioabsorbed. So structure is so important, so important. So just try to keep that in mind. It's going to come back. It's going to come back. Those of you who want to go into your RN, you're going to take pharmacology. Isomer is going to come up. And I seriously doubt that someone is going to take that time and explain it in the way that I did. I hope I am wrong. Let's get toward the end part of this thing. We have to get some processing from the retina, right? We have to have, I keep saying this, data. They don't use data, but I'm saying data. Photoreceptors in bipolar cells generate only graded potentials. Graded potentials, not action potentials. That's what they're saying there, the photoreceptors in the bipolar cells. So when light, don't worry too much about this. Hyperpolarizes photoreceptors, don't worry about. They stop releasing the inhibitory neurotransmitter glutamate. We've talked about that neurotransmitter so much, glutamate. Oh boy, it always keeps coming up. Bipolar cells depolarize and then they release the neurotransmitter onto the ganglion cells. It is at that point that the ganglion cells generate action potentials transmitted in optic nerve to brain. You gotta, it has to go back to the brain, guys. And now we're seeing the process, the information processing, or as I like to call it, data processing. This is a little bit of light, kind of going back a little bit to what we talked about. Light adaptation. So when moving from darkness into bright lights, we see a glare. Why? Well, because the rods and the cones are strongly stimulated. So, and then large amounts of pigments are broken down instantaneously, producing glare. That's the reason why we see that. Dark adaptation. When moving from a bright light into darkness, we see blackness. Why? Oh, the cones stop functioning in low light intensity because they can't function in low light intensity. That's the job of the rods. So bright light bleach the rod pigments, so they're still turned off, and the pupils then begin to dilate. Make sure we go over this one here in terms of light blindness, condition in which rod function is severely hampered. Well, why? We just said that the rods are of light sensitivity, meaning that means they don't need much light. So you're probably going to use them at night, right? Well, therefore, if you use the things that are responsible for that light, if you have light blindness, are probably damaged somehow, some way. So therefore, that's what it's trying to say, condition in which the rod function is severely damaged. Some of the rods are damaged or degraded somehow, some way. So it says ability to drive safely at night is impaired. There you go. There's the word, degeneration, due to rod degeneration. Now, real briefly, this is very important for this case study so you can start grasping some concepts here. We get into B12, vitamin A. What is one of the reasons for the degradation commonly caused by prolonged vitamin A deficiency? Gotta keep telling you that, guys. I keep on saying that vitamin A is going to help you make that retinol. It's retinol is being synthesized from vitamin A. But as all things, if you figure this thing out soon, if administered early, vitamin A supplements restore function. What a cool thing, right? But I wish I could just go on and on and on and on and just talk to you about vitamin A and retinol and its functions. That's the stuff I really enjoy. Continuing on the pathway, visual pathway to the brain, we know we have those axons of retinal ganglion cells from optic nerve, right? So we have that, medial fibers from the eye cross over at the optic chiasma, then continue to the optic tracts, which means each optic tract. Let's see. Okay, yeah. So then you have to contain fibers from the lateral aspect of fly on the same side and the medial aspect of the opposite side. And then you have each carries information from the same half of the visual field. Mainly this right here, guys, these 2 bullet points. I don't think I asked you much about depth perception. I really don't think I did. That could be wrong, but I'll just briefly say it, what it is. Both eyes view same image from slightly different angles. So we have a visual cortex fuses these slightly different images, resulting in 3-dimensional image, which leads to a depth perception. Well, if we have problems with our cortex, then we're going to have issues with depth perception. This is going to require input from both eyes to create the depth perception. If you have just, if you're, as it says here, requires input from both eyes, right? If you just put one hand over, it throws your depth perception off a little bit. This is what I was trying to say. Loss of an eye or destruction of one optic nerve eliminates the true accuracy of depth perception, okay? So keep that in mind. A stroke can affect the left visual cortex, which leads to blindness in right half of the visual field, okay? So that is the end, okay? That is the end, and that's what I wanted to focus on, the eye. Now, the ear, I'm gonna cover some of that. I'm gonna cover that in lab. Briefly, I'm gonna go over the ear, but I really wanted to just spend a good amount of time on the eye. Again, you know why, right? Not trying to rhyme here, but you know the reason behind that, and the reason behind why we're spending a lot of time on the eye is a question in your exam, right? Again, since we are in lecture, you will have special senses, a Pearson exam, right? Now that we've covered this, I'm going to get it uploaded into your Canvas, and then I'm going to open up your exam, your lecture exam. I don't remember how many questions there are because I didn't see a feature that was tracking questions. Again, I'm always 100% honest. I don't know. I was clicking on the ones like, yeah. It gave me a test bank. And I'm like, okay, yes, this topic. But I went through every single question you had. They gave me, I think it was 100 and I think like 90, maybe 190 something test options. Of course, we didn't go past 100 questions, but those are the options. But I went through each and every single question for you. And I did my best, again, to select content that we really went over. So please, just because it is a different format, it's a different type of test, don't let that build any types of anxiety or test-taking inabilities, right? It's just a different platform. We do need to be very adaptive in today's world of how we take exams. When you take exams, you could do like an NCLEX, you can do your TEAS exam. They're going to be different platforms, different companies hosting it. Things are going to look different. And unfortunately, in today's world, we have to be very fast at moving and adapting to the tech. So this is just a simple exercise of a different platform to use. That's why I wanted to use it. And it also, again, like I said earlier, makes me feel good that I'm using different types of resources out there and the students get to look at different things. Not my main go-to, again, ladies and gentlemen. Pearson is not my main go-to, but I wanted to give you a little look-see into that. All right, well, I'm going to pause it and open it up to any questions to save the duration of the video. So everybody watching this video, have a wonderful day and we'll see you soon. I'm going to stop the recording.